Hello, welcome to Pen with Love. I am Tutor Additional.
For the Neco 2026, the specimen for the biology practical is what we want to address now. And like I used to say, please note, this channel is not in any way support examination malpractice. We encourage you to look into what we do here, practice 2026 Neco biology specimen analysis. These are the specimen given to us. Specimen A, pelvic girdle of a small mammal. Specimen B, cervical vertebra of a small mammal. Specimen C, the caudal vertebra of a small mammal. Specimen D, pawpaw, a pawpaw fruit. Specimen E, guava fruit. Specimen F, quill feather. Specimen G, agama lizard. Specimen H, a small mammal, either rat or rabbit. Specimen I, quadrat. Specimen J, sweepnet. Specimen K, housefly. Specimen L, pride of Barbados fruit or pride of Barbados pod. Specimen M, cashew nut. Specimen N, millipede. Specimen O, crab. Specimen P, sorghum green. Now, these are the specimens given to us, specimen A to P, and we are going to be looking at each specimen diagram and analysis. What are the possible questions that can come on each specimen and comparing two or more specimens together? I will go into area of concentration. Based on this specimen given to us, we can concentrate our analysis on arthropod, classification of arthropod. When you look at the housefly, millipede, crab, they are classified under phylum Arthropoda. So they are arthropod. We are going to look at questions under that. Then we are going to look at aspect of reptile versus mammal, and we are going to look into their comparison, their differences, their similarities. Then the bone given to us, those bones given to us are from mammals. So we are going to look at the vertebra. Another thing that we should be expecting is the ecological instrument, the ecological aspect in biology, which include the specimen like quadrat and that of sweep net. Other thing we should look into the fruit aspect, which include specimen D and specimen E, and I think specimen M. Then finally, we are going to look at some diagrams that we may be asked to draw, and we are going to give tips on drawing in biology. I believe if this is not your first time, if you have been here in this channel before, you will have a clue in the drawing aspect. Nevertheless, for the first timer, we are still going to look into tips to follow when drawing in biology. For specimen A, specimen B, and specimen C, they are under the same category. So let's look at them one after the other. These are the pictures of the specimen that is going to be given to you. Specimen A, the pelvic girdle of a small mammal. Specimen B, the cervical vertebra of a small mammal. Specimen C, the caudal vertebra of a small mammal. It can be of a rabbit, it can be of, but you cannot know. So just tell them small mammal. So what are the likely questions that can come under this? Number one, they can ask us to state one function of each of the specimen A, B, and C. Remember, they can also ask you where those specimen can be found. So the pelvic girdle can be found in the hips, while the cervical vertebra can be found in the neck region, while the caudal vertebra can be found in the tail region. Now, let's continue. Like I said, apart from identification and asking of the region, they can also ask you to state function. Specimen A serve as attachment of hind limbs to the vertebral column. Specimen B, it supports the head and permits neck movement. Specimen C, it supports and moves the tail. It supports and it moves the tail. So those are the functions of specimen A, specimen B, and specimen C, in case you are asked. Another question that can come under this is observable differences between specimen A and specimen B, or between specimen B and specimen C. Now let's look at under specimen B and specimen C as we have it in front of you. Specimen B, there is large vertebral canal, while under specimen C, small vertebral canal. Under specimen B, it has transverse foramen, while under specimen C, no transverse foramen. Under specimen B, broad neural spine, while under specimen C, reduced neural spine. When you look at specimen B, they have short central, while under specimen C, they have long central. You yourself can look at how close specimen C and specimen B look like. In specimen A, there is no neural span. Ileum is present, while ileum is absent in specimen B or C. In specimen A, there is no neural spine, while in specimen B, there is neural spine. Another thing they can ask is to talk about specimen A adaptive features. What makes it adapt to that position of joining the hind limbs with the vertebral column? So we can gladly have the following adaptation. 1. Strong and rigid bone for support. 2. Broad surface for muscle attachment. C. Socket for articulation with femur. So these are the 3 observable adaptive features that we can talk about. Specimen D and specimen E. Under specimen D, they will give us a transverse section of pawpaw fruit. And looking at the diagram, the one showing cutting section of this particular fruit is what is going to be presented to you. Likewise in specimen E. And this as the transverse section or cross section. No, so it is not the longitudinal form that we are regularly familiar with, especially when we are cutting this fruit at home. The questions that can be asked under this are as follows. A. Apart from the identification of the fruits which we have mentioned, they can ask you which type of fruit are they. Both specimen D and specimen E belongs to berry fruit. Then they can ask you which type of placentation did they exhibit. Both also exhibit axillary placentation. Both exhibit axillary placentation. They can also ask you to classify the specimen B, specimen D and specimen E. Let's look at the classification one after the other. You can see on your screen, we have kingdom, division, which is also known as phylum. We have class, we have genus, and we have species. And if you are in any way asked the scientific name or botanical name of each of these specimens, you should give them the genus and the species name. And In a previous video on this channel, we've explained that anytime you are writing a scientific name or botanical name, you are to write the genus starting of the genus with capital letter and the species with small letter. Because you cannot write in italic form, you have to underline both genus name and the species name. Mind you, you are underlining separately. Now, purple belongs to plant A under kingdom, belong to division Magnoliphyta, belong to class Magnoliospida, then genus Carica and species Papaya. In a nutshell, we can say the scientific name or botanical name of purple is Carica papaya. Under guava, it is classified as follows: kingdom, plant A; division, angiosperms; class, Magnoliospida; genus, Psidium; and species, Guajava. So, in a nutshell, we can say the scientific name or botanical name of guava is Psidium guajava. Another question that can come under this is observable differences. You know, those fruits, when you look at their fruit type, they are the same. When you look at their presentation, is the same. So they can ask you observable difference. And when we talk about observable differences, one of our videos, we've stated that observable differences means what you can see. You can see with your physical eyes, not what you think, not what anybody say. So, under papaya, the differences we can see is that under papaya, large cavity, while under guava, there is small cavity. Under papaya, numerous black seed concentrated centrally, while under guava, numerous small seed embedded in pulp. Under papaya, soft fleshy mesocarp, while under guava, firmer pulp. Now, they can also ask you to give similarities between the two. They are both fleshy food. They both belong to berry. They both contain many seeds, and they are both edible fruits. So these are the similarity you can make mention of. Another thing we can look into under this is agents of disperser. Agents of disperser. So agents of disperser for the both are still the same. Animal can be their disperser, and mammals, animals like bird and some other things like that. Then man too can be their agent of disperser. Now let's look at the economic importance of the two specimens, specimen D and specimen E. Both are very, very rich in vitamins. They can be eaten as food. They are also used in juice production. So these are the likely questions that can come under these two specimens. Now let's move to the next specimen. Specimen F, specimen G, and specimen H. Now, classification can come in. Specimen F is queen feather. Specimen G is agama lizard. Specimen H is rat or rabbit. They can give you any small mammal. It can be rat, it can be rabbit. So anyone given to you in your school, please note. Now, they can ask you the following questions. The adaptation of queen feather for flight. They can ask you where can we find, which animal can we find queen feather. Queen feather can be gotten from any bird. So they can ask you to classify bird, and they can also ask you that which class. We are going to talk about that one too. Let's look at adaptation of queen feather for flight. 1. Lightweight. 2. Broad vein for air resistance. 3. Strong ratchets. Then smooth interlocking barbs. These are the things that allow it to be able to what? to adapt to flight. Now they can also ask on adaptation of agama lizard, which is specimen G. Scaly skin reduce water loss, strong claw for climbing, well-developed limb for movement. They can also ask us the differences between agama lizard and rat or rabbit, the small mammal given to us. When you look at the screen, you will see the difference I've highlighted. Under agama lizard, we have covered with scale, the body is covered with scale, while under rat, the body is covered with fur or hair. Cold-blooded animal, they are poikilothermic, while rabbits are warm-blooded animal, which are homeothermic. Then agama lizard lay egg, they lay egg, while rabbit or rat, they give birth to their young ones. That means lizard, they are oviparous, while rats are viviparous. Then agama lizard has no mammary gland, while rat has mammary gland. They can also ask you to classify agama lizard specimen G, rat or rabbit specimen H, and they can ask you from which kingdom or phylum or class does the animal in which we can get quill feather. So quill feather is gotten from bird, so it belongs to Animalia, kingdom Animalia. Bird belongs to phylum Chordata, and they belong to class apes. And that is why I stopped there, because these are the questions they can likely ask under that. But let's look at the classification of rat and agama lizard. If you are given rat, fine. If you are given rabbits, you know, you are going to do classification of rabbits. Agama lizard and rat or rabbit, they all belong to kingdom Animalia. They belong to phylum Chordata, and also bird too, Animalia Chordata. Now, agama lizard belongs to class Reptilia, while rat belongs to class Mammalia. Agama lizard belongs to Agama, and the species name is also Agama. So, scientific name for lizard is Agama agama. While that of rat, the genus is Ratus, while the species is Ratus too. That means we can say Ratus. So let's look at the next specimen. The next specimen we are going to be looking or analyzing is specimen I and specimen J, which are for ecological aspect. Specimen I is quadrat, while specimen J is sweepnet. So they can ask you to tell us what they are used for. Specimen I, which is the quadrat, is used in estimating plant population, and specimen J is used for collecting insects. They can ask you, how do you use quadrat? If you want to use quadrat, the following steps are to be taken. Throw quadrants randomly. You count the organism within the quadrat. Repeat several times, and you are going to calculate average population density. And they can also ask you why quadrat is used for plants. The reason is for that is because plants are stationary, they don't move, and that is why quadrat can be used for them. Unlike insects that we use sweep net in collecting, they can also ask you which type of insects can we collect the sweep net with, that specimen J. Then you can make mention of grasshopper, butterfly, and some other insects that you can remember. Then they can also ask you what are the precautions while using sweep net. The following are the precautions while sweeping net, using sweep net. While you are using sweep net, you have to sweep gently. Avoid damaging organism. Then empty content carefully so that you will not kill the organism you have caught. The next specimen we are going to analyze now is specimen K, specimen N, and specimen home. Specimen K is housefly, specimen N is millipede, specimen O is crab. You can see the diagram of these specimens on your screen. Apart from identification of this specimen, you can be asked to classify them, especially to the kingdom, the phylum, and subphylum. Specimen K, N, and O, the three belongs to kingdom Animalia. And likewise, the three of them also belongs to phylum Arthropoda. Therefore, the only difference we have there is under subphylum, and which is the main thing they can ask as the question. What are the subphylum of the three specimens? Which are specimen K belongs to subphylum Insecta, specimen N belongs to subphylum Myriapoda, while specimen O belongs to subphylum Crustacea. So another thing they can ask is comparison of all these organisms. Common characteristics: they all have jointed appendages. That is, they have jointed legs, they have segmented body, and they all have chitinous exoskeleton. All the three possesses exoskeleton. Now, let's look at comparison. Housefly and millipede. Housefly has body division of head, thorax, and abdomen. That is 3 body divisions. While millipede has 2 body divisions, which is head and trunk. Now, under housefly, another difference we can bring up is the pair of wing that is present in housefly, while millipede has no wing. Likewise, crab has no wing. So they can ask you the economic importance of crab. It is used for a source of protein, source of income, and it's also used as food. Then they can also ask you the life cycle of housefly. First, this is exhibit complete metamorphosis. That is from egg to larva, from larva to pupa, and from pupa to adult. So it is a complete metamorphosis. They can ask you that too. Now let's proceed to the next specimen, which are specimen L, specimen M, and specimen P. Specimen L, Pride of Barbados fruit or Pride of Barbados pod. Specimen M, cashew nut. Specimen P, sorghum grain. They can ask you which placentation does specimen L exhibit. It exhibits marginal placentation. They can also ask you what are the mode of dispersal of each of the following specimen. Specimen L, explosive mechanism, when it explodes into the air. Specimen M, animal or man. Specimen P, wind and man. These are the mode of dispersal of those 3 specimens. Let's look at more questions that is possible to be asked here. They can ask you to classify sorghum grain. It belongs to kingdom Plantae. It belongs to division Angiosperm. It belongs to class Monocot. It belongs to genus Poaceae and belongs to species sorghum. So the botanical name for sorghum grain can be Poaceae sorghum. Then, what is the food storage in sorghum grain? It is starch, carbohydrate. Economic importance of the specimen, the 3 specimens. Specimen L, Pride of Barbados, it can be used for ornamental plant. As ornamental plant, it can be used as edge plant. Specimen M, cashew nut. Cashew nut, the nut can be eaten as food. It also serves as a source of income. And specimen P, sorghum. It can serve as food for humans and it's also feed for livestock. They can ask you to find aiding dispersal. What are the things that is aiding the mode of their dispersal? Pride of Barbados pod, it has dry pod. It also splits explosive when ripened, and that is why we say that mode of dispersal for Pride of Barbados is explosive mechanism. What aids this explosive mechanism is because of the dry pod and because it splits explosively when ripe. Then when you look at cashew nut, the cashew nut, we said the mode of dispersal is by man or animal. When you look at it, what are the things that aid it? Attractive fleshy stalk. It has an attractive fleshy stalk and it's also edible fruit attract animals. For sorghum grain, it is small and light for wind to be able to carry. It's also produced in large number and that is why man to take it for food, and from there it can be dispersed through man. And we have come to the end of the analysis from specimen A to specimen P. The concluding part now is the drawing aspect. The drawing aspect. I want you to go and study the diagram of specimen A and specimen B very well. This is the diagram you are looking at, you are seeing on your screen. Specimen A, specimen B, specimen C, and specimen D. These are the diagram you are seeing on your screen. And mind you, when you are drawing, all this drawing now, like I have promised, very soon we are going to have a comprehensive video on biology practical. I'm working on that and the video will be dropped very soon. So when you are drawing, make sure you are drawing and label on a side, label on one side. So study the diagram of specimen C and specimen D very well too. Also study the diagram of specimen F. Specimen F, as I'm looking at that specimen F, there will not be much question on it, so the diagram might be very important for you. So specimen G2, a gamma lizard, you can study that too. Then specimen I, sweetnet, and specimen K rather, which is housefly. Study the diagram very well too. Also study the diagram of specimen L, the Pride of Barbados plant, fruit rather. In WAHEC this year they gave us pod, granite pod, and in the exam they were later asked to open up the granite pod and draw what they have. So study this diagram too, is very important. The question might come from that angle too. And on the right hand here on your screen, you have the tip for few tips for drawing in biology. So please kindly study this tip very well. No shading in biology drawing, no artistic drawing, but we need simple drawing. Drawing title is very important. Drawing title is very important. Always label in one side of your drawing. Erase neatly. Label line must not cross each other. Do not allow your label lines to cross each other. Maintain uniform thin line when you are drawing. Never use pen at all while you are in drawing aspect. Always draw to the given dimension. I explained this. If you have not watched if you have time, you can also take your time in watching the WAHEC session, because there are some specimen too that is still here. I explained clearly that when you are drawing, when you are given 8 to 10, your drawing should not be less than 8 and it should not be more than 10 in terms of longitude, that is how long it is. So also, don't forget your magnification. If you are given hand lens to magnify it, if it is times 2, if it is times 4, so indicate in your drawing so that you're going to have your full mark at the end of the day. So we've come to the end of the class. I wish you the best of luck in your exam. Please like this video, comment, share, and don't forget to subscribe. Share with your friends that are writing this year Neco this week. By the grace of God, I wish you success in your exam.